from, oops, let's take off the graphic so you can see us. Where the hell is it? From Micro TV, this is TWiEVO, This Week in Evolution, episode 104, live streamed on July 24th, 2024. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Nels LD.
Hey, Vincent, great to be with you from LD Lab Studios, as you said, in Salt Lake City. And actually a state holiday here in Utah. This is Pioneer Day.
Really?
Celebrating the somewhat complicated history of settlement around these geographical parts. So the campus is empty. It's like the university is closed, but the podcast, the live stream goes on. And then later this evening, wildfire smoke and fireworks will be mingling together as the locals are celebrating. Actually, it's interesting, Vincent, the sort of Gentiles, as we call ourselves, the non-Mormons in in Utah, we've sort of created a splinter holiday on Pioneer Day called Pie and Beer Day. And so that's an event that's gained steam to celebrate living in a state with sort of a pretty strong religious heritage that's constantly changing and progressing in interesting new ways.
Are you going to go to this celebration, Nels?
Nope, I'm going to lay low and enjoy kind of the quietness on campus, get in a few work items that are lagging behind, a few deadlines, and then escape into the mountains as things heat up here down in the valley and just enjoy a summer evening.
Sounds good. Here in New Jersey, I am today. 77 Celsius, 77% humidity,
Ooh.
27 Celsius and cloudy. It's a little humid out, but not very warm. I'm here today because I have clinical update at 5 p.m.
Yeah.
And then it's not enough time to get home and do the live stream at 8 to 10. So I decided just to stay here.
Gotcha. You're at Racaniello Home Studios, it looks like.
Yeah, this is where, I set this up during the pandemic, right?
Yeah.
Got a good light, a good camera, a good microphone, some stuff behind me. That's a Michelle Banks virus.
That's great.
Collage there.
Yeah, I love it.
And it works. The tech is pretty good here, so I like that. Let's see who is here today. Tell us where you're from. I'm going to introduce our moderators.
Great.
Who keep civilized this small group.
Easier said than done.
Here. Yes, Les is here from California. Andrew is from New Zealand.
Great.
Barb Mack is from the UK. And there you go. Three Les, Andrew, Barb Mack. Three mods. That's probably enough for 20 or so people.
Yeah, and it will probably warm up the engines as we go here. Thank you all for being here and navigating live stream traffic.
Let's see who else is here. So Barb Mack says it is rainy, overcast day from England, 17C.
Warm and dry here in Salt Lake. Wildfire season has emerged.
ME is from Helsinki. Sorry, I missed you. I was in Helsinki for 2 days at beginning of June. And there it's nice weather. 24C now, but it was much cooler than that when I was there. Helsinki was a nice town. Somebody else on the live stream, Wednesday night live stream, is from Finland. I'm sorry, that's Sweden. They told me somewhere to go south of Stockholm, Helsingborg. They said it's a great walking town, Helsingborg. Have you been to Sweden or Finland?
Yeah, absolutely. I haven't been to Finland. That's on my list as a growing sauna enthusiast. I just built a sauna at my house. But yes, I lived, Vincent, for a year in Stockholm.
Nice.
This was a while ago. I was 2 years old when I arrived and 3 when I left. So the memories
I don't know if you remember anything.
A little bit foggy, but I've been back many times over the years. One of my favorites is, so first of all, in Stockholm, the Vasa Museum.
Yeah, the Vasa's cool.
Did you make it?
I was there before.
Okay. Oh, yeah. Yep. Yeah. That's right. And then, if time is short, getting on a local ferry to just kind of dot the archipelago and hit all the small islands, grab lunch or a beer at a small location, or with a little bit more time, taking a longer ferry through the Baltic to Gotland, and then getting out on some bikes on that island and padding around there is pretty spectacular. And this is the time to do it. This is the time of the year to do it where there's plenty of light available.
So I was in Stockholm in the end of May, just before I went to Finland, and we went to see. We went on a boat trip over through the locks.
Nice.
There's still some locks that can actually go from one end to the other. To the, I don't remember what bodies of water on it. One is the Baltic, I guess, but I don't know what's on the other side. And we took about a 3-hour lock trip on this lovely boat. You know, you move into a lock, the doors close, it fills up, then you go. It's so cool.
Yeah, I love that.
Cool.
Yep.
That was a lot of fun. But
Nice.
I have a friend there, Agnes, who, you know, says anytime you're coming, we'll show you around. So she said next time we have to go to the archipelago.
Yes, I agree. I endorse that one. Yep. Did you get in a sauna when you were in Helsinki, or didn't have time?
Oh, I didn't. No.
Okay.
But you know, the funny thing is, when I was in Finland, I was at a meeting 3 hours north of Helsinki. Jyväskylä was the name of the town. So university town. We were at a conference center, and there was another conference across the way on. It was a sauna conference. Everybody was very happy at that conference.
Yeah, now we're talking.
Okay, let's see here. So who else is here? Jan is from Tucson, 32C.
Great. Yep, sounds about right.
Jan says you get Pioneer Day in Tucson, we get Rodeo Day.
There we go. Similar energy. Yeah.
Jeremy is from Seattle, where Nels used to hang out.
Absolutely.
Right?
Good. Welcome, Jeremy. Good to have you back.
Abdul Rahman is from Colorado Springs.
Great.
Looks like at a meeting, SNIS 2024. Dominic is from Poolsbo, Washington.
Beautiful part of the country. You can get around by ferry in and around there as well. Although the Washington State ferries have been a little beleaguered with repairs and delays and things like that, but still, again, in the summer, one of the most beautiful parts of the world.
Fields of Green is from Salinas, California.
Awesome. Welcome.
It's pretty nice out there, too.
Mm-hmm. And agreed.
Abdul Aziz is from Saudi Arabia, if I remember. Claire is from the UK. Elizabeth is from West Virginia.
Awesome.
Michael is from Rotterdam, where I've been. I was there in 2019. Lovely meeting.
Nice.
Michael is from Southwest Pennsylvania.
Great. Welcome.
Abdul Aziz, thank you for your support of science communication. We appreciate your support of our programs.
Thank you.
Dominic says, sauna enthusiast is my favorite phrase.
Well, I'm just getting out of the blocks. This is new territory for me, even though I grew up in Minnesota. And the sauna lifestyle is alive and well in and around Minneapolis. I can report to that. But I just fired up my electric heater for the first time. I have to still finish a little bit of the sauna room before I can really get in there. But the heater from Finland is here in Salt Lake, and it's running hot. So yes, I'm growing that that's my new hobby, my mid-year's resolution.
Andrew says, I think it was Denmark where they found the best Viking longship underneath a museum dedicated to Viking longboats. That's pretty funny.
Yeah, and the Vasa that we're mentioning at the top there, this is a Swedish warship and its maiden voyage sunk in Stockholm Harbor and then got dredged up hundreds of years later, and is now this great sort of testament to human hubris and humility, in addition to being a really cool cultural artifact. Yeah, one of my favorite museums in the world.
John is tuning in from work. More interesting than background music here in Eden Prairie, Minnesota.
Minnesota, welcome, John.
Another
Great
place
to have you
where
along.
you hung out for a while. Actually, you were born in Minnesota, right?
Born and raised, and all the way through undergrad in a small college in Northfield, Minnesota. And then I hung around and worked for a few years before I went to grad school in Chicago.
So
Yeah.
the ancestry of your family
Hmm.
is Nordic, right?
Yeah, mostly Swedish,
Swedish.
tracing back 3 or 4 generations, right? 4 generations. And then with some Norwegian heritage mixed in, about a quarter in that direction. Yeah. Although I haven't done, have you done the 23andMe or related
I
things,
have, yeah.
Vincent? Yeah.
I
Yeah.
have.
I've never done that. I've kind of,
I'm mostly Italian, but
yeah. Yeah.
some other stuff thrown in there.
Mm-hmm.
Let's see. Tom is missing this one tonight. See you tonight on live stream, 8
Ah.
p.m. Eastern is office hours. My guest tonight will be Michael Schmidt.
Oh, cool. That sounds
All
great.
about bacteria.
Tom, no sweat. Thanks for checking in, and we'll catch you the next time.
Joseph is from Ontario, Canada. All right, that'll do it.
Welcome.
As always, folks, if you have questions during our discussion, we have an interesting paper. It's a pretty woolly paper today.
For sure. Yeah, this is going to be fun. And don't hesitate to be liberal. Questions early enough, and it might take us a minute to catch up, and we'll really do the roundup probably closer to the end, depending on our sort of how heated we get or carried away we get in the conversation here. But let's dive right in. So the title of the paper today, Vincent, is Three-Dimensional Genome Architecture Persists in a 52,000-year-old woolly mammoth skin sample, pushing the field of ancient DNA analysis, ancient genomics forward in a big way. This just came out in the July 11th issue of the journal Cell. And let's run down, if you've got it up on the screen there, let's run down a few of the authors. There's a great, in addition to the graphical abstract, there's a great cover to the issue itself. It's this cartoon of the back end is a stone part of a woolly mammoth, and then the front end, it's sort of coming out of the stone and seemingly coming to life, which sort of echoes on their ability to get really high resolution look at the genome of this 50,000 plus year old sample. So the first authors, there's 4 of them. 4 first authors
So
in the
many authors
arms race
paper.
of that's right.
Look at them all.
Big group, and I think that reflects the work here. In order to gain meaning out of DNA from a sample that's 52,000 years old, you need a diverse team from, in this case, around the world, from Copenhagen to Houston, from Sweden to Spain. And so the first authors, Marcela Sandoval Velasco. Second first author, Olga Dudchenko. Third first author, Juan Antonio Rodriguez. And our fourth first author, Cynthia Perez Estrada. I almost feel like I'm announcing like the stars of a sporting event, or starting a lineup or something like that. And that's, I think, really the case here. Senior authors, 3 corresponding senior authors, Marty Renome, Thomas Gilbert, and Erez Lieberman Aiden. And as I said, international consortium, so we can see some of the places here. The Center, Center for Evolutionary Hologenomics at the University of Copenhagen in Denmark, Copenhagen. Center,
Hagen. You were right the first time. It's Hagen.
Yeah, Hagen. Okay, thank you. Yeah. This is like the highest stress part of the live stream, is trying to pronounce the locations and people's names.
Nels, I used to say Copenhagen, but Paleo Schechter lived in Denmark for many years, and I used to say Copenhagen all the time. And he said, finally, once he said, Vincent, you don't have to make it German. It's Copenhagen, not Copenhagen.
Pulled you aside. Maybe I'm thinking of the snuff, the chewing tobacco, Copenhagen, from back in the day. Anyway, Center for Genomics in Barcelona, Spain, Center for Paleogenetics, Stockholm, Sweden, the Swedish Museum of Natural History, Stockholm University. Now flying over to the U.S., the Department of Genetics, Cell Biology, and Anatomy at the University of Nebraska Medical School, Northeastern University in Boston, Texas, in the mix here, and with some important contributions. I could keep going and going, but time is of the essence. So what I will say is there's a great, and what kind of got me onto this paper was a great Twitter feed from one of the first authors, Juan Rodriguez. And in this thread that he put up on Twitter, gives some of the backstory of the paper. And one of the tidbits here is took them 9 years to do this study, I think, from having the samples to building out the tools to analyze the genome. And in particular, the sort of advance here is that as we look at up until this study, a lot of ones, samples from Neanderthals or Denisovians, as we think about human ancient history, or even other woolly mammoth samples, et cetera, are, usually involve a bone fragment, either from the teeth or I think the ear bone. We've talked about this, Vincent, on TWiEVO before as it relates to Neanderthals and Denisovians. And even if in those well-preserved bone samples, the DNA is highly fragmented, and so you're getting really short, just little chunks of the genome that you're trying to distinguish between. Is this actually part of that ancient genome? Is it contamination, which is a massive issue in the field? And then taking those small snippets and really just cobbling them together into a genome, using a modern genome as a template. So you have no opportunity to sort of take longer stretches of DNA and think about how that's organized at the chromosomal level into the genome, all of the nucleic acid or DNA in each of our cells, the nuclei of our cells. So here, what the authors, it's really in kind of right out of the gates, I would say, it's really in how that sample, it's a skin sample from a woolly mammoth, was preserved. It was dehydrated. And this is huge. So they draw the analogy of, sort of like beef jerky. That process of food preservation or dehydration turns out to do really good things. If you take away the water, as this thing is even at room temperature or plus or minus, or in the cold, if it's the sample, for example, is from Siberia, because of the way, just by chance that it dried out very early after the death of this female woolly mammoth, that had some really favorable results in terms of preserving the structure of the skin down to the cellular level, and then even within the cell, the subcellular level, the nuclei. And then what the authors work on here or present here is an analysis of the chromosomes and the ability to get to that level of sequencing in genome organization. So I don't know if they've coined this term, if it's out there before. I'm a little outside of my field, but subfossils, fossilized chromosomes, Vincent. I love this. Yeah. So because they think they have some of the structure in place, then what they can do is not just sequence snippets, but try to get at chromosomal organization, right? And so this is that 23andMe again. There are 23 chromosomes. The elephants have 28, at least the modern elephants. We don't know how many what the chromosome number or karyotype is of mammoths, and the authors will present that in a little bit. But to just get us up to speed, so the technique they're using here, it's called High-C, which is actually sort of a name that captures like previous techniques and then putting it at high throughput. So the original sort of approaches here were 3C, or capture chromatin conformation technique. So let's unpack that a little bit. So chromatin is sort of, our DNA to fit all that DNA, those 3 billion base pairs that we encode in each of our cells. You really have to pack that down, because if you just put that physically, if you just put that molecule, that DNA, stretch it out, I forget, there's, all these comparisons, or you can stretch it around the world or something like that. And so proteins, histones, and other components are organizing the DNA, packing it really tightly into what's called chromatin. And there's sort of several layers of this packing that occurs, so you can get all of that information into a really small place, the nucleus of the cell.
So we have a question from Les, which basically you have just answered, but
okay. Yeah.
How does 6 feet of DNA string fit in a tiny cell nucleus?
Yeah. So thank you, Les. So that's the 6 feet. Imagine getting that into a cell. You need a microscope to look at, and so that's where this chromatin structure comes in. And there's layers of this packing in order to pull it off. And so there's been almost a cottage industry over the last decades of techniques to better understand the organization of that packing, and then at each of these various levels to sort of describe the geography of the genome in a sense. How are, what are are there sort of reliable locations of DNA material in the chromosomes as it relates to the boundary of the nucleus. And it turns out there's a lot of, organization and patterning in there. It's not random. So a lot of the genes that are sort of along the length of the chromosome aren't physically in 3D space next to each other. They're next to each other, but that's not the whole story there. There's a lot of, folding and looping. And so even regions or genes in different parts of the genome or on different chromosomes are often closely physically associated with each other, even though they're not along that sort of 6-foot stretch of DNA if you put it all end to end close to each other. And so the idea then is that there's been these techniques, High-C, so it tries to capture that chromatin conformation, something about the location of the genes on the chromosomes with each other. And then High-C is just a method of doing that at high throughput. So 3C, you could pick one location, one gene in the genome and say, okay, what are your nearest neighbors, using some of these tools, this biochemistry, some of these tricks. High-C comes in and says, okay, let's not just pick one location in the genome. Let's do this genome wide for all of the chromosomes and get a sense of that 3D organization. Okay, so that's what they're up to. And, honestly, I think, Vincent, no one had really tried this for ancient samples, at least as far as I know, because it's so fragmented and you just assume that all of that organization is gone. And so that's kind of, I think, underlines really the pioneering part of this effort here on Pioneer Day in Utah is to, try to see. And they spent a lot of time and effort, and I'm guessing a pretty substantial part of these 9 years to try to see if they can really pull this off, and then to do a number of kind of complementary analysis to see if there's in comparisons to see if what they're seeing might reflect what was happening in those genomes, in those cells, in those nuclei 50,000 years ago, frozen in time, and then available for us to see for the first time as it was unearthed or dug up from the Siberian permafrost. Okay, so how do the DNA regions physically associate in 3D space? So we talked a little bit about chromatin. Here, the biochemistry they're using is a cross-linking technique. So you actually take in that cell that you've processed a chemical that links the DNA together. Then you finally, for the first time, blow it all apart, cut it into pieces as needed, although there's already a lot of fragmentation here in the nucleic acids given the length of time. And then you ligate together the interacting parts that have been cross-linked, and then put them, process them in a way that you can sequence them. And then the output of this is you get a lot of chimeric DNA. So meaning that you're kind of walking along the genome, there's one gene that you'd expect, and then all of a sudden it jumps to a different chromosome, a different set of genes. And because you've sort of frozen this or captured it using those techniques, what that tells you is that those disparate locations of the genomes, maybe even on different chromosomes, are physically in close proximity to each other in that organized 3D nucleus.
What does ligation mean, Nels?
Yeah, so ligation here means that you're using an enzyme to take two fragments of DNA and to ligate them together. And so the DNA has been cross-linked, which can be a covalent connection, but then you're going through some processing to ligate it into another piece of DNA that allows you to read out those along the length of these chimeric or sort of Frankenstein-like pieces of DNA that reflect, again, that sort of close proximity. This also, Vincent, gives you a sense of not just sort of the geography of the genome, so to speak, or the morphology, but also the activity of the genome. So there's kind of two flavors of chromatin, or sometimes referred to as the epigenetic states of the DNA around there. And, so basically dividing, as I think simplified, is to dividing it into what's being actively transcribed, so-called open regions of the chromatin. So there, in that case, the histones will reorganize or get out of the way a little bit. That allows these other proteins, the transcription factors, to come in to transcribe the genes into RNA that then gets translated into proteins, the sort of, workhorse, the central dogma of molecular biology. So there's actively transcribed regions, or euchromatin, as it's called, versus these packed away or less active or inactive regions called heterochromatin. And using the High-C and some related techniques, they're able to say something about that, and then again draw comparisons. And we'll kind of come to that in a few minutes here as we think about, what kind of meaning can you get out of cataloging the geography of a genome, especially as you're kind of comparing these ancient samples or this ancient woolly mammoth sample to the modern elephants, and asking, okay, well, what happened in those 52,000 years of evolution from the Siberian cold to Africa or Asian modern elephants today?
So now, as you talked about, you cross-link.
Yeah.
So
Yep.
we have two pieces of DNA that are close together in this high-order structure, this chromatin, right? So the cross-linking will link them, right?
That's right. Correct.
And then when you sequence, you'll sequence one, and then you cross over to the other because of the cross-link. So that being able to detect that means you need to have a genome to map it to, right?
Exactly. And so that opens, and you put your finger on the biggest question here, which is, okay, you can do this even on a degraded sample, but are you really getting something that reflects the organization that's out there? Or is, has time just come in and moved everything, mixed everything up? And so that's actually where the paper, I don't know how, we won't go deeply into it, but uses some ideas borrowed from physics. So glass transitions that would really predict that this, by dehydration, and just how that process unfolds relatively quickly, that once this is dehydrated, if that's happened sort of in an organized or orderly way, that you'd get this glass transition where everything is locked into place. And so there's still, time and insults, et cetera, will wear on the sample, but that is really sort of the key idea here, linking it to some of the physical principles where they think they're onto something in terms of getting this level or this resolution of information from these ancient genomes. Okay, so we talked about High-C. They developed, this isn't sort of as straightforward as taking a sample from like any of our cells or another, like an animal, a modern animal. And so they put a lot of effort into and introduce a new technique they're calling Paleo-High-C. And they want to address three questions, as they've modified this technique to accommodate for this ancient sample. So we'll go through their three questions that as they outline them, and give them kind of a scorecard here as we go, previewing what the answers are. So what extent is the 3D topology of the genome preserved? And they're going to try to argue that's pretty good. That's going to be based largely on comparisons to the modern elephants. What can you learn about the biology of an ancient species based on this kind of new level of information? And so they'll lift up or highlight some intriguing differences compared to modern elephants. So it's sort of this balance between comparing where you're hoping to see some shared features that you'd expect, but if you're confident that the differences are real and not just artifacts or part of the decay process, then can that teach us something about the differences in the biology if we sort of turn back the clock 50,000 years? And so they find a few genes that they think are interesting differences in terms of were they active or inactive in the chromatin level analysis. It's also, it turns out, I think this was kind of lucky, the flip of the coin here, this was a female woolly mammoth that they sample that they're using. And so that allows them to think a little bit, as we think about genome organization, one of the massive topics here is the sex chromosome. So that in the case of mammals, the X chromosome, so females have 2, whereas males are XY. And so if you have 2 X chromosomes, this and twice the number of genes on the X chromosome compared to males, this raises a challenge, which is dosage compensation. If all the genes are expressed or transcribed into RNA, translated into protein at twice the level, that can lead to some real issues biologically. And so X inactivation, or the turning off into that inactive state, the heterochromatin of half the genes basically on the X chromosome, or X chromosomal inactivation, which happens at sort of a more chromosomal level, they're able to look at that and see some interesting differences in other mammals, humans and mice, compared to elephants and mammoths. Third question is, how can this chromosome structure persist for so long? And here again, borrowing back or adding in some of the physical principles and thinking about glass transitions and freeze drying will sort of be an interesting part of the work here. And I think, thinking about the impact, in terms of the biological meaning, I would say, relatively That's probably maybe the least strong part of the paper, doing a little podcast peer review here. But but the the technical advance, and to really be able to get this level of information out of a sample this old, I think is really, what got me excited about it. I think is getting other scientists excited. It sort of makes you rethink what's possible and how to prioritize, as samples, as archaeologists, paleontologists, et cetera, are gaining access to new samples all the time. What are the ones that are sort of worth it to put this level of work into? Okay, so the skin sample itself was from the head of a woolly mammoth. This is, the Pleistocene period in Siberia, 50,000 years ago.
A nice picture.
There you go. Yeah, so that elephant, the woolly mammoth, is dying, freeze drying, and then all the way up to today. And then, so the question is, or the comparison is, the assumption would be shown on the left there. So that the 3D geometry, that's the circle is the nucleus, and all the colored squiggles are the chromosomes, is that the geometry would be lost versus preserved. And so those boxes with the red color, that's the High-C and related analysis, sort of zooming in at these different levels of organization. So where are the chromosomes sitting in the nucleus? And is there sort of an organization to that, or are they all just scrambled all over? And so that diagonal red line, there's a lot of plots like that that indicate. So on the y-axis are all of the chromosome coordinates, and on the x-axis are all the chromosome coordinates. How are these things related to each other? If it was all simple, just kind of stepping along, what you would see is chromosome 1 would match with chromosome 1 for proximity, 2 with 2, 3 with 3. But when you see some of the off the diagonal increases in red color in these plots, that's indicating some of this higher order compartmentalization, and you can see that even at this cartoon level. They dig in deeper. So actually, Vincent, if you scroll down, we can see the microscopy here shows that, this, I think, gave them some confidence to go ahead with the work. So panel B is actually someone's gloved hand holding the woolly mammoth skin. And then panel C shows some microscopy here. And so you've got the kind of expected layers of the skin, the epidermis and dermis, underlying it, just from kind of classic microscopy histology. And then zooming in farther, now we're looking at a single cell, and it sort of looks like a kernel of corn sort of shriveled up, and not that sort of round sort of sphere that you might expect before it was dehydrated. But that probably reflects the loss of the water and other sort of hydration in the sample, but then holding everything in place there. So they have a few stats there at the bottom. So that, so they took some of these shriveled grapes, these nuclei, so to speak, and went ahead and did that High-C approach. I think they did it 26 times with the samples they had. And the modifications they made it, so it's the reason it's Paleo-High-C is that they did a couple of things to kind of try to stack the deck in their favor for robust analysis. So they immediately preserve that sample in the field. They use ethanol, and that's a tip of the cap to the microbes out there that are constantly contaminating these samples. And in fact, the vast majority of what they end up sequencing and doing High-C on are microbial genomes, and that sort of comes with the territory of ancient genome work. The second modification is that they try to sequester the chromatin, some of these structures, the DNA wrapping around the histone proteins. They use beads in order to try to lock that in place. And then finally, they actually don't do what you usually do if you're doing High-C or these chromatin capture techniques. They don't shear or chop up any of the DNA, because it's already sort of, nature has done that for you ahead of time. And then finally, all of the library amplification, again, they're doing this in, really clean room level, beyond clean room level laboratory setups in order to try to avoid more contamination. I think we talked about it in the early ancient DNA sequencing days. Some of the first reports of Neanderthal mitochondrial genomes were actually those of Svante Pabo, the researcher who was developing these techniques for the first time. It was, a true and sincere attempt, but the issue of contamination is real, and there have been tremendous leaps forward for both trying to reduce that, which is impossible, but then even as you get contamination, to sort of identify that and to take that away from the analysis pipeline as they go forward.
In this panel D, just
Yeah,
so that people know,
yeah.
the beads are those little bluish things connected to each strand.
Yeah, that's right. And so you can see sort of marching from left to right that they're putting them in ethanol, cross-linking and grinding. And then so basically the cross-links are made. And then at that point, they're trying to still hold, like a lot can happen just in the processing. And I think given the fact that these are already fragmented, that's where the bead treatment comes in, is to kind of try to hold things together. Because then once it's cross-linked, you're not done. And as you can see, they've kind of have it cartooned here. So they have two, they're in blue is one chromosome, and orange or brown is the second, that they've cross-linked together. They're proposing that these are, or they think they're identifying based on the cross-link that these are sort of close together in location. And so there's further cutting that goes on, and there you can see the ligating of the ends.
Right.
And so that's the step to basically get it ready and then from there you purify the DNA. It's already in pretty small pieces. It's been chopped up. So that's what the beads are doing, is just to try to give it a little more stability. And then at that point, once they've purified it, they pull it down. So they get all the DNA away from all of the enzymes and the buffers in that test tube. And then you've got what they're calling a paleo-friendly amplification and sequencing. And what you can see, so what we're talking about with this chimeras is now, because of that cross-link, blue is now ligated to brown, and you sequence across there. And based on that boundary, you can say, okay, this part of the chromosome is located close to this part of this other chromosome. So 4 billion letters later, they've got a lot of data to sift through. And in fact, most of it is contamination. So I think if I'm looking, so only like something like almost 98%. So only 2.5% is not contamination based on that sort of filtering out. So you're comparing sequences to other microbes, bacteria, probably mostly some viruses, of course. And so they end up going from 4 billion to 115 million bases remain. And even more low efficiency of those, the ones that are useful for doing that kind of chromatin capture, considering those chimeric, the geography of the chromosomes, only 0.1% of the samples have those kind of long range contacts. And if you do this with, so just to kind of illustrate how hard the sort of degree of difficulty in this project, if you took a modern elephant sample, and they do this to compare them, 40% of what they generate is actually useful relative to this 0.1%. So, they're really trying to push the technology here in order to deal with this still pretty highly degraded sample. So 5 million useful reads at the end for considering how the kind of morphology of the genome. But then, of course, the question is, okay, well, of those, how many are real? Are you just, has nature taken its course and you're sort of fooling yourself into thinking that these are connections? Because how do you distinguish sort of what's real from what could have happened? So here what they do is, some statistical analysis, some power law scaling, and they use those modern elephant samples where they do that, where they get 40% and they're able to kind of see that. And then they ask, if you do this kind of statistical treatment, do you see similar patterns that you'd, kind of at that 30,000 foot level that you'd predict between an ancient sample, even though there's been 50,000 years of evolution? These are still, a mammoth looks a lot like an elephant, and probably the genome also at that kind of submicroscopic level in terms of its organization looks the same too. And in fact, that's what they found. So that kind of is, the evidence from that analysis consistent with the idea that those locations of the chromosome within the nucleus were kind of locked in place at the time of death of that mammoth. And so then they want to go farther and do kind of classical genomics, right? Assemble the genome. Again, you can't quite do that. They don't have enough information here to do that de novo, which would be like wildly cool if you could like do that without a reference. But they use the modern elephant references, also some of the other ancient mammoth genomic efforts that have been done over the decades to do what they call reference assisted assembly of the genome. In order to kind of convince themselves that they're not just overfitting the genomic analysis, they sort of test drive this. They take sort of veer off here into another mammal. They take the donkey genome assembly and they make it more sparse. So they basically take away a lot of the information and modeling or mimicking what they're able to get from that ancient mammoth species, and then use the horse genome, which is roughly as different as maybe the 50,000 years genetically speaking. And then they also get pretty similar outcomes doing that. So they've kind of convinced themselves both by comparing to the kind of patterns that they're seeing from modern elephants, and then doing this sort of assisted assembly with donkeys to horses, which is sort of a similar distance of mammoth to elephant, that they're actually putting together something that reflects kind of real life 50,000 years ago.
Nels, what's amazing is that when they did the sequencing of the woolly mammoth, they get 98% of the genome.
Yeah, and so that's right. And so here they're going back. So we just talked about how things got kind of whittled down, but then they go back and do regular old style shotgun sequencing. And exactly, they got 98% of the total.
They got all 28 chromosomes.
Exactly. And so that's another, that you put your thumb on it. What is like kind of one of the exciting things for the genomicist is that we've never been able to predict the karyotype or the chromosome number. And doing the analysis here, they come up with 28. Turns out elephants have 28. So that's, maybe not super surprising, but the fact that you could do that kind of starting from scratch with that sample is, again, one of the cool things here. Okay. So given that they now have 98%, they've done the assisted assembly. Are these sort of, using this catalog they have now, or this first glimpse into the chromosome organization, what's going on here? So, and again, sort of the useful functional analysis is to consider those two types of architecture. So or actually this is a little bit different. So there's, when people do this 3D reorganization or look at the organization of genomes, there's sort of two types of patterns that emerge from this. One is that within a chromosome, the central part or centromere of the chromosome and the telomeres, the end of it, are sort of close together in space. This is called type 1. The second type of architecture, type 2, is where the between chromosomes, or the chromosomes are sort of packaged. You had that cartoon up a while ago where it's in, you have like these colors are chromosomes, and they have like their own sort of locations, their own zip codes in a sense in the nucleus. And so now they do these what they're calling plaid plots. There, you just zoomed by one, which is fine.
This one?
Yeah, that works. So here, this is at the chromosomal level. So again, on the y-axis, you can see these are single chromosomes on the y and x-axis. The diagonal indicates that as you go along, it's sort of business as usual. The plaid plots are starting to define that a little bit of the organization, so blue and red. And this is showing kind of the red stuff is sticking together, the blue stuff is sticking together. And what you're looking for are sort of two patterns. One is more like in the upper left for chromosome 10 of the elephant, or in going across. And then in the woolly mammoth, they're looking at a few different chromosomes here and seeing that the blue and the red are sort of interspersed in this almost plaid-like pattern. And that's consistent with this type 2 architecture that you've got, the chromosomes sort of have their own zip codes. This also allows them, so starting with, as they kind of take off from that and sort of go to the next level of organization, the chromosome loops. So they have sparse data here, but again, they see patterns that are matching pretty closely to the modern elephants. And so, I think if you had a specific hypothesis about genes in these regions or how they're being regulated or co-regulated, this gives you sort of a roadmap to think about how they're organized and how this might be working. You can do this now with the modern elephants and then add in the sort of a new channel, which are the woolly mammoths, and try to make a prediction about how the chromosomes are organized, how the loops are there, and how this all fits into the sort of growing knowledge about how genes are regulated, or chromosomes are regulated during the cell cycle, et cetera, what genes are turned on when, which also depends on the tissues. You have sort of these patterns that start to provide some information. As I mentioned before, maybe one of the most interesting chromosomes to look at is the X chromosome, right? The sex chromosome, based on the fact that we know a lot about the patterns of inactivation versus activation as it relates to the gene dosage, turning off, so to speak, half of your genes if you're a female with two X chromosomes versus the males with the XY. And so here they spend a lot of time sort of digging in, and they make a big case about, well, first they identify, what looks like bar bodies. So that's inactive, like major inactivated regions of the X chromosome, very similar for modern species. The fact that they can see this means that they're now at roughly 50 nanometer resolution in order to see these kind of features. And that's after 50,000 years, which is, again, I think pretty impressive. And so one of the differences here is that, so the way that the X chromosomes are inactivated is commonly referred to as bipartite inactivation. There's sort of super domains in the X chromosome, or boundaries between active and inactive chromosomal regions. And that's different for the woolly mammoths, which they define as having 4. I think that result's interesting. It kind of fell maybe a little bit flat for me in the fact that modern elephants are 4 as well. And so they haven't really said anything new, I would say, about the evolution of this. Maybe they've looked at that for the first time, and that indicates that this persisted in the lineage leading up to modern elephants and is different from mice and humans. I think where it gets...
Elephants and woolies are not that distant, right?
No, they're not. But that's where, but that gets to I think the more interesting question right, Vincent, which is, okay, well, these are pretty similar. 50,000 years isn't that much time for a mammal, given generation times, population sizes. So what's different? And so here they use another sort of fancy pipeline for genomic analysis, another analysis tool called CRUSH. And if scientists are just so vulnerable to making up, acronyms, right? So CRUSH stands for Compartment Refinement for the Ultra Precise Stratification of High-C. And High-C, if you unpack that, is this 3C, but at high through anyway. So it's like the nesting doll model of genomic techniques here. But basically, this is a way to compare active and less active regions at 50 kilobase resolution. So we've moved from sort of nanometers when thinking about the looping of the chromosomal structure to 50,000 base pairs, letters across there, and to define or to make a prediction about what's going to be active, what are gene regions that are being transcribed, made into proteins, versus ones that are less active, the heterochromatin. And so here they do this CRUSH technique, both on their catalog or their analysis of the mammoth compared to the modern elephants. And they find about 820 genes in about 1,400 regions of the genome where things look different. And so now I think we're getting a little bit, sinking our teeth into, potentially a little more biological meaning here. So they have a few examples. I'll just do one in the interest of time. But it's the EDAR gene. I think we talked about the EDAR gene. It's one that's under strong selective pressure. I think we saw it related potentially to the plague, the bubonic plague, Yersinia pestis. There is some work by Luis Barrero and others that lifted up this gene in some ancient samples of humans. But here, the EDAR gene, so this is involved in hair follicle development, as well as having some involvement potentially in cold adaptation, as well as potentially some immune-related things. But so elephants have an interacting gene that is active, maybe an inhibitor of EDAR compared to mammoths. And here it's sort of fun to draw a line. It's a little bit speculative, but to draw a line to how this is the woolly mammoth, a very furry creature living in Siberia, compared to the modern elephants that have lost most of the hair, living in more temperate climates. And, does this sort of explain some of that difference, is that you have this potentially inhibitory gene active in modern elephants, but not in the mammoths. It sort of like takes off the brakes on losing your hair or not developing those hair follicles. Another kind of interesting. Oh, so and then they do a little bit of a deeper dive into X chromosome. They do a couple of those. I think still early days in terms of then following up with experiments that might get deeper into the biology here. So then to kind of close it out, Vincent, they circle back. It's sort of I'm not going to bluff here. It's a long paper. I think they, and fair enough. It took 9 years, like 50,000-year-old sample, that international consortium of people you want to, kind of celebrate and really tell the story here. And they do that. It's a long paper. But they finish kind of, returning to how is it that this sample was so well preserved compared to most of the bone samples, et cetera, where, you can't get anywhere close to this kind of level of genomic analysis. And so now we're talking about food dehydration again. And things get a little bit wild here, I have to say, in the experiment. So, they use the beef jerky analysis. Maybe it's a little bit more like prosciutto or Serrano sample, or even dried cod, I think is how the food preservation would work here. But they do an experiment with beef. So they take fresh beef and either just let it kind of degrade, where even in short amounts of times, you lose all of the cellular structures, et cetera. I think contamination, as the microbes come in immediately, get into the game, versus doing the dehydration, right? And so this food preservation, which limits the microbes, and I think turns out from a physical standpoint actually preserves the structure not only of the cells but of the nuclei and of the chromosomes, those subfossils. So they don't go for 50,000 years, they go for 96 hours and 1 year, and then do High-C to compare. And actually in those beef samples, even the 1 year to fresh, they see something very similar. And so, from here, they want to actually kind of torture the beef samples a little bit to see if they, how kind of resilient is this? It's because you don't have 50,000 years. Well, let's try to stress out the sample. So I'm going to read a direct quote from the paper, which sort of catches you a little off guard as you're kind of sifting through all of the technical details of the high technology. They say, strikingly, chromatin architecture in a year-old dehydrated beef was robust to many perturbations, like getting run over by a car, immersion in acid, and being pulverized with a shotgun. So if you look at figure 6, they actually have cartoons of these interventions where they took their beef samples, ran it over with a car, dipped it in acid. I think they even have one on the right there. They had a former Houston Astros baseball pitcher throw a baseball at it. I have no idea why they chose this, other than, it looks like they're having a lot of fun in addition to sort of bringing the field forward here. This might have also been, as I said, it's a long paper. Maybe this is like a test. Are you still reading the paper this far into it? So anyway, so you can see their 1-year-old sample. Each of these plots, they're just doing kind of a high-level High-C analysis, and you can see from so you've got the beef sample fresh, you dehydrate it 1 day, you go for a year, or 96 hours, you go for a year. And then the year's old one, they shoot it with a shotgun, run it over a car, throw a fastball at it. And those plots are similar. The, signals they're seeing, those dark red dots compared to the lighter ones, are basically identical. And so that's different than the lower left, where this is the one where they don't dehydrate it, let it go for 96 hours. And you can see that you've got a lot of kind of random noise. The dots are just spread out. The dark dots are spread out throughout, different than the chromosomal organization on Y to X axis than you see from the preservation or the fresh sample. So here, I think in a kind of fanciful way, they're really trying to underline that dehydration works, whether we're doing this for food, for our beef jerky or prosciutto, et cetera, or if you're doing this for maintaining these samples or collecting samples, what to look for. And actually, I don't know if they kind of circle back to that, but so skin samples that are probably on the surface are maybe your best bet here, moving away from the ear bones or the teeth. Or the bones might be more soggy. This is kind of me just speculating, but the skin, depending on where the animal or the sample passes away, if those environmental conditions are just right, that you could get this kind of preservation. So I'm guessing it's in the deserts in Arizona or in and around Tucson, or even here in Salt Lake in the valley, where you'd have a better chance at natural dehydration relative to the sort of wet and humid climates from, ancient history, 50,000 years ago, plus or minus.
They have. Speaking of prosciutto, they actually mention it in the discussion. They say they're talking about dehydration and how effective it is at preserving. They say that's why dehydration has been used for thousands of years to make products like prosciutto legs and chugno stable at room temperature.
Yeah, exactly. And so, two benefits here, both is to limit the microbes, but then also to save the morphology of the cells, of the nuclei of the chromosomes. So, I've given some so so one more couple of loose threads here to kind of close it out as we're coming up on the hour. So, and this felt right at the end, sort of the last paragraph of the paper. They throw in kind of another comparison, which really felt like this is probably a real peer reviewer's comment. It's like, okay, really cool, but this is all based on one sample, right? And so is this just sort of the magical sample and kind of a one-off? You did it this one time, but what are the prospects for, being able to apply this technology as we identify other samples? And so they actually take another sample from a woolly mammoth, one of the best preserved samples. It's not as old. I think it was 39,000 years old is the estimate from when it died. And try that Paleo-High-C and see what they get. And they just very quickly, just sort of skim over, oh yeah, it looked roughly the same. And so that's great, actually. I think, probably a lot more work hidden in that reviewer comment, but I think it just speaks to that this isn't a one-off, that, given everything that they saw, all the work, the 9 years of work they put into the first one, that there's some real kind of, possibilities for extending this work into other samples that are also well preserved, that we already have collected from other species, or as new ones emerge, or to help kind of even guide where to look for some of these samples going forward, if this is what you're hoping to do.
This one has a name, actually.
Yeah, yucca or something like that. That's right. There's not as ancient genomicists team with museums and archaeologists, paleontologists. I think there's, it's definitely a small community, and there's limited samples, like in hitting everything just right in terms of the preservation is really rare. And so these are really precious samples.
The main subject of this paper, though, didn't have a name.
No, that's right. Yeah.
Yak-inf. That's the closest. Yeah. So that's interesting.
Yeah, agreed. Finally, the last kind of piece I have here is, and one of the things that Cell Press does is, in the discussion, they have a section called Limitations of the Study. And actually, this is a kind of a breath of fresh air, because in science, we're so, we're always trying to convince our peers or editors that the work that we've done is like really special and great and best thing since sliced bread kind of thing. But to step back and have a little bit of honesty about no scientific study is perfect. There's holes, there's limitations, and to lift that up and to highlight that, I think is really, the right way to go, to kind of a little bit of honesty about the challenges or the limitations of interpreting this. And so they have a nice section there where I think they, kind of measure that, highlight that the data they recovered is sparse. Comparisons they did, I think they give it some credibility or credence, but that this, kind of proceed with caution or take it with a grain of salt. It's really, in order to get this information based on those samples, that's really the advance. But the idea that they've really hit the nail on the head here, or that you can squeeze that kind of information out of a sample like that, is just probably impossible. And so, I think, I'm really glad to see more and more studies include that as they get published, to not just lift up the sort of positives, but also the limitations, to give sort of an honest assessment there, or closer to one at least. But take-home message. Yeah, go ahead.
Go ahead. That's all right.
Oh, I was just going to say, take-home message for me: these dehydrated samples have really good promise.
I wonder how old you could go if you had a dehydrated sample from older, right?
Yeah, great question. And so that's where I think they just kind of tacked it in at the end. But is that 39,000 year sample better than the 52, or was it preserved differently, right? To kind of get at some of those parameters that could really, could you, and I agree, Vincent, can you push this back 100,000 years, give evolution even more time to kind of have that time machine energy to some studies going forward? I think, stay tuned. Obviously, I don't think this is the last word on dehydrated samples and ancient genomic analysis.
I wanted to show people some of so this is the methods section here of the paper, right?
Yeah.
They talk about how they do things. And, I'm just scrolling through, there's a lot of text, but then you get to a part that is mathematically very intense here, as you can see, right? Where
That's
they
right.
have to make various models and assumptions and so forth. It's really a lot.
There's a lot. And I know it. And that gets into that, connecting it to that glass transition and some of the physical, like the physics and the predictions there. Exactly. That's what goes into some of those power calculations that they do.
I love this square root
equation
here. Look at that.
Come at you with a lot. You can sometimes get overwhelmed by by the math. But that's, again, I think that's one of the, to their credit. And as we go through the 35 different footnotes based on the 30 or so different institutions, those connections all the way from the museums, from, Trondheim, Norway to Barcelona, Spain, to Stockholm, to all across the globe, to Nebraska. I mean, that's what is really a fun part of this work, and to see that to come forward like that. Yep.
Let's see what we have here back in the comments. So since we dropped off, Vanity Nutrition has shown up. Thank you.
Oh, great.
One of our moderators. Good to see you.
Yeah, welcome.
And then also FSM is the dog from Wales. Thank you for coming. And FSM says, so we can clone a mammoth.
There we go. Yeah. So are we getting closer to that? So that's a whole nother field. With some proponents, Beth Shapiro, Santa Cruz, George Church at Harvard. I'm still a little skeptical. And what we mean by clone, so, they're proposing that using both the Paleo-High-C and the shotgun sequencing, they've got 98% of the genome here. 2% is a lot when you're thinking about the size of the genome. So I don't think we said it. So I think that 98% accounts for like 3 gigabases of sequence, 3 billion base pairs, not unlike our own genome. But so 2% is a big number, and there's probably a lot of important information in there. So when we take, sort of the cloning of mammals, stretching back to Dolly the sheep, sort of another one of these sort of technical steps forward. But when you take that high degree of difficulty there, with the high degree of difficulty here of even cataloging what that genome looked like, I don't think we're too much closer to that Jurassic Park moment where the dinosaur is fogging the Jurassic Park Jeep window or whatever. But there is a move in de-extinction, probably closer to reality, where it's not actually resurrecting or cloning the ancient species itself, but it's doing genetic engineering in ways that might sort of allow a modern species to adapt either to climate change or something closer to the habitat where it came from before. And so I think there are efforts, mostly privately funded, going in that direction. And so that's probably not quite Jurassic Park, but as close as we're going to get. I think in our lifetime, we're not going to see woolly mammoths. We might see some hairy elephants, but I don't think we're going to see actually woolly mammoths like the one that was there 50,000 years ago. Yeah.
So Andrew says there's a video from GeoGirl on bringing mammoths back from extinction and why it's a good and bad idea. So there's a YouTube link.
Oh, thank you. Great. Yeah, good to have that resource. And this is the reason, it kind of captures your imagination for obvious reasons and raises some really important ethical considerations as we do advance the technology as geneticists, genomicists, genetic engineers. When and where should we apply these kind of resources? And so that's where, the arguments about de-extinction and should we put this level of resources into that versus like just habitat restoration, the kind of simple tried and true ways of supporting populations. And that's pretty important conversation to have. Yeah.
FSM says the scientist in me is curious, but the ethics are horrific.
Yeah, so like agree. So but I don't think that's any different than technology as we kind of trace back our history as a species. I don't think we've invented anything new here. Maybe our tools become, and become more powerful. The genetic tools. We're kind of, I think, at a new chapter in gene therapy as an intervention for things for genetic diseases, things like cystic fibrosis, sickle cell, where there's been some advances recently. And I think we will see some more there. But you're right. I mean, the ethics here, the ethics have been here all along and are still here. Maybe to me, the thing that's most alarming is as a society, as a country, as we've gotten more and more focused on STEM, we've maybe de-emphasized the humanities, right? And the value of those careers. And that's where the ethics questions I think are the same, but maybe the number of people pursuing that, the philosophy majors, right? The people working in like we need that. The historians, all the information we've gathered, that information, and there are experts in it. But as we sort of de-emphasize or let go of that, that's to me really the danger here. Yeah. Yep.
All right. So what I learned today as a question, as the DNA will have exons and introns, how will the scientists be able to find which region provides the protein? How is it different from the current elephant to the mammoth protein?
It's great. Love it. Yeah. So those, again, so actually just doing these sequence comparisons. So we've been kind of flying a little bit higher at chromosome level organization, but as you come down to the actual gene sequences, exon by intron, you can make those comparisons too. They're not going to be perfect. So, 98% is sort of the average here, but it's all kind of fragmented and chopped up. Yeah. But those 50,000 years isn't that long. And so, a lot of that gene by gene sort of architecture, exon, intron boundaries, it's all there. And that's where, I think this resource for scientists who are interested in specific genes can go back and start to draw those comparisons potentially more powerfully for the first time. There's a lot of other mammoth genomes out there, so you can start to build multiple comparisons in there and really get at that level too, where you can understand, at a gene level all the way to a chromosome touching another chromosome level in the nucleus, that whole kind of range of information coming into focus for the first time. Yeah, great question. Good point.
Here is Rima from Iowa. Welcome.
Yeah, good to have you back.
Dominic says 50 nanometers at 50,000 years. Pretty good resolution.
Yeah, agree. It's wild to think about that. Yep. Hard to wrap your brain around it. Yeah.
Dominic also says 100% support this level of play,
Yeah, or they're running over their sample with a car. I agree. And, I think that happens more than we let on. I give the authors credit for, not being too serious here. There's plenty of playful scientists out there, but it doesn't always show up in the printed page or on the, PDF file of the work. And so, a big credit to the authors for probably what I'm guessing, among those first authors, there's a little bit of that playful energy coming out. Yeah. Good for them.
All right. Nels, you want to do some picks?
Yeah, that sounds great, Vincent. So, speaking of kind of mysterious science, my pick of the week, I can't, we're talking about elephants, so I have to go back to the microbes or the viruses in this case. So I've got a Twitter thread, that caught my eye recently. It's mysterious SARS-2 variants showing up in samples from sewage, sewer samples. So there's a guy on Twitter, I think his handle is Solid Evidence. He analyzes a lot of fecal or wastewater samples looking for viruses coming up. This is Mark Johnson at the University of Missouri. So he has a really cool, he's kind of trolling through the massive and growing data set of SARS-2 variants, and he found one in Utah. So I guess maybe my pioneer day, all the way to the SARS-2 variants. There was in a town called Hurricane, spelled hurricane, but pronounced hurricane, in the south of Utah, near a lot of the national parks. Maybe a visitor came for a few days. And based on and he built a tree. So all of the SARS variants are kind of organized by the old classics, the alpha, beta, gamma, omicron. But there's a long branch, which there's, I think, 3 samples that show up, and Mark Johnson steps through sort of his detective work on asking where did this come from and what does this mean? So it only showed up 3 times in Hurricane, Utah, or in and around there. He's postulating that it was an individual maybe a visitor to the national parks or whatever, who had a SARS-2 case, but it doesn't look like this thing has spread. But it's got literally hundreds of mutations never seen before, just in this individual who probably had a GI tract version of SARS-2. And so the fact that we're able to kind of make these sightings based on wastewater samples and kind of do this detective kind of epidemiology on the public databases at the genetic or genomic level, I think is really fun. So that's my pick of the week, a mysterious SARS-2 variant in Utah.
So we think this came from some patient somewhere?
That's the idea. So, so he's sort of taking what he's calling the Utah variant.
Yeah.
And this was back in early February. And so you can compare all these sewer sheds basically.
Yeah.
And then, so you see where the wastewater is, and Hurricane doesn't have a hospital, but nearby, I think St. George does. And so then there's more information there, not all the way to the patient level. But what's wild is, 100 private mutations, meaning that of the 30,000 or so letters, 100 of them are ones that have never been seen in all of the millions of samples that we've looked at. And so, yeah, he's kind of, the mysteries is, was someone on vacation, looks at the hospitals nearby and, kind of comes, why would someone have been tested for COVID a week apart and have both samples submitted for sequencing from a random city with no hospital? And that's the mystery. You'll probably never know. And nothing to worry about. It doesn't seem like this variant is going to take off the way Omicron did, right? But it shows, this is kind of the reason I think this is great for TWiEVO, and we'll have the link for the pick if you want to do a deeper dive here. And follow Mark Johnson. He's doing these kind of, detective sort of situations as he's scanning samples coming up from time to time. But all this evolution that happens, but then doesn't catch on, that's the majority of evolution, right? And then we just sort of put in the spotlight, whether it's a mammoth from 50,000 years ago or, a really successful lineage that wipes out the rest of a virus lineage, in this case, a SARS-2 lineage. Those are the things that we really focus on, and not the sort of ones that don't make it. But it's all out there, and it's interesting to weigh that from time to time. So anyway, that's my pick of the week. How about you, Vincent? What's your science pick of the week?
So I have a curious article from Science. Cocaine sharks found in the waters off Brazil.
Wow, another mystery here.
This is by Eric Stokstad. And basically, scientists in Brazil report that 13 sharp-nosed sharks from coastal waters near Rio de Janeiro had traces of cocaine in their muscles and livers. This is published in the Science of the Total Environment. And there is cocaine in the environment. It's been detected in sewage and rivers in many different countries, probably from labs that process it, right? But this is concerning because the fish are taking it up, obviously, and we don't know who else is taking it up and what would be the effect on the fish, or even some people who eat sharks or other fish, right?
Yep.
100 times higher concentrations than found in other aquatic creatures. Apparently, there are packages of cocaine floating around in the oceans as well.
Oh, yeah. And the sharks are finding them, it looks like. Yep.
Cocaine pollution.
Cocaine sharks. It's right out of the sort of movie genre there. But I think that kind of connects to what we've been talking about, right? Is like, how do animals interact with their environments in ways that are unexpectedly influencing their biology? Wow. I had not heard about the cocaine sharks.
Well, then, what was I going to say? Yeah, you want to know what it's doing to the sharks, what it's doing to people who eat them, but also the extent that it's being picked up by other animals. And it's an example of how, we're changing the environment, and it's not just us, but the whole environment suffers.
Exactly. Right. I think there's a lot of, interest, rightfully so, in how much in addition to cocaine, but how many pharmaceuticals, right, that get into waterways, whether someone doesn't finish a prescription, or how you dispose of your prescriptions, your prescription drugs, and how that, could be influencing life around, aquatic life in particular, which is in constant contact with the water that we're changing as it's being, as chemicals are showing up in
Couple
drugs.
of comments here. Yeah. FSM says, Sagan warned us about the increase of scientific illiteracy, and you warn us of ignorance of humanities. Yeah, that's absolutely right.
Yes. And I mean, don't get me wrong, I'm all in favor of resources towards STEM, but how about STEAM, where we include art, humanities, and really go for a holistic look at how the work of understanding the meaning of life around us at all resolutions, how we as a species use that information and apply it. Yeah. Thank you, FSM. I think we're on the same wavelength here.
Andy wants to know, aren't sharks incredibly indiscriminate eaters? I don't know.
Well, that's a question. Are they seeking out these packs of cocaine is my question, Vincent.
Or if it's in the water, I don't know, maybe they're concentrating it, right? May not be eating anything, but so anyway, interesting article, interesting observation. And
yeah, didn't see that one coming.
That, folks, is TWiEVO number 104. I want to thank all of our moderators for coming by today. That includes Vanity Nutrition, Barb Mack, Les, and Andrew. Thank you so much for being here. Thanks everyone else for being here also. We will have at 8 p.m. tonight, office hours, another live stream. And my guest will be Michael Schmidt, so check it out.
Very cool. And adding my thanks as well. Appreciate it, everyone. Enjoying the conversation as we live stream together.
You can find TWiEVO at microbe.tv/TWiEVO. It's where you'll find links to things we talk about. If you enjoy our work, we'd love to have your support. You can go to microbe.tv/contribute right up above me. That's the link. Or you can go to Venmo above Nels.
Trying to get that. There we go. Yep.
But essentially, Microbe TV is a 501(c) nonprofit entity. So in the U.S., your donations would be federal U.S. tax deductible, and we need your donations to continue. If you like our science coverage, please support us.
Yes.
Nels LD is at cellvolution.org. Early Bird on X. Thank you, Nels.
Thank you, Vincent. Great to be together as always. And happy Pioneer Day, everyone.
I'm Vincent Racaniello. You can find me at microbe.tv. Music you hear on TWiEVO, let's turn it on here, is by Trampled by Turtles. You can find their work at trampledbyturtles.com. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next week. Till then, stay curious. Bye-bye.